Assalamu alaikum. In this lecture, we will learn about the remaining MCQs related to testing of biological molecules. So let's start. Diastase is an enzyme that breaks down the starch into maltose. Right? Maltose is a reducing sugar. A sample of the starch is treated with the boiled diastase and left for 15 minutes. Whenever it will be treated with the boiled diastase, then it will convert starch into maltose. Right, now we have maltose in our sample. Sample of this mixture was then tested with the iodine solution and then with the Benedict's reagent. So whenever we will test it with the iodine solution, we will turn the blue. We will get the blue-black color, because not all the starch has been converted into the maltose. Some of the starch is still present in the sample. And when the Benedict solution will be given and the sample will be treated with the Benedict's reagent, then the color of the reagent will not change, because maltose itself are non-reducing sugar, and non-reducing sugars do not give any color change with the Benedict's reagent. So option A here is the right answer. A student carried out the test for the biological molecules on the same sample of the milk. The test and the results were as follows. Heating to 80 degrees centigrade with the Benedict solution give a red color. Yes, this gives. Boiling with the acid follows by the neutralization. Then heating to the 80 degrees centigrade with Benedict solution also gives a red color. Yes, it gives, because all the reducing sugar, non-reducing sugars are converted back into the reducing ones, that is the monosaccharides, and they all are reducing sugars, so they give the red color. And biuric solution gives a purple color. Yes, it gives. Adding the iodine solution gives a yellow color. No, adding the iodine solution gives a blue-black color. So which kind of biological molecules will be present in the milk? They can be the reducing sugar. Non-reducing sugars are not present, because we have converted back them into the reducing ones. So we will say that we have only reducing sugars. And proteins are present, because we are getting the biuric test here and getting the accurate results, so proteins can be present. Reducing sugars are present, and starch is not present, because the iodine solution do not give blue-black color. So option C here is the right answer. 4 solutions were tested with the Benedict solution, and the table shows the color of the solutions after testing. First solution gives green color, second gives blue color, and the third one gives brick red color, and the fourth shows the yellow color. As we can say that how much the quantity of the reducing sugars present in the sample, the more intense will be the red color, and the more it will lead to the color transformation more rapidly. So let's check. Which row shows solutions that can give this result, right? So solution 1, if the solution 1 contains 0.05% sugars, solution 1 is giving the green color, which is the initial one color change. For example, blue to green, and then green to yellow, and then to the red color. So whenever if we will say that only a least amount of the reducing sugar will be present, then it will give the green color. So least of them is that one, and the blue color will appear when there is very low amount of reducing sugar or non-reducing sugar will be present. So we can say that if there will be the 0.5 non-reducing sugar, then it can give the blue color. Yes, it can. And 1%, so we have to find out the least amount of the non-reducing sugar, so it is this particular one, 0.5%. Solution number 3 gives brick red color. It means that it has a huge percentage of reducing sugar. So among these, 1% is correct one. So now we will go towards the solution 4. It means that the least amount of less amount of reduced sugar is present. So among these, 0.1% reduced sugar is found to be the correct one. So option A here is the right answer. A solution of amylase was added to the suspension of the starch. After 30 seconds, the 3 samples of the mixture were tested with the iodine solution. Benedict's test or with the biuret reagent. Whenever the iodine solution, we will test the sample with the iodine solution, then it will give the black color, because not all the starch has been converted into the simple sugar. Some of the starch is still present in the sample, so it will give the black color. And when it will be treated into the Benedict solution, it will not directly show the red color, because a few amount of the reduced sugars are present in it, because the sum of the starch has been degraded into the simple sugars, not all the starch. So it will give only a intermediate color, that is the green color, not directly red color. So whenever the biuret reagent will be tested whether the proteins are present or not, so it will give the purple color. Some kind of proteins, means as we have added the amylase, so it can also show the presence of the amylase, so it will give the purple color. So option A here is the right answer. Which student carried out a Benedict test on the several different known concentrations of alpha glucose? Which graph represents the result correctly? Whenever we have the different concentrations of the glucose, we will put the concentrations of glucose on x-axis and the time to color change on the y-axis. This will be the accurate order. So whenever the more the concentration of the glucose, the less time it will take to get a color change. So in the graph 1, option number A, we can say that the more the concentration of the glucose, the less time it will take, and whenever the concentration of the glucose becomes lower here, then the time will enhance. So we can say option number A here is the right answer.